Good morning. My name is Nicole Mittman, and I'm the Chief Scientist and Vice President of Evidence Standards here at CADTH. Thank you for joining us for this CADTH workshop series on platforms, baskets, and umbrellas. You'll hear a little bit more about what we're talking about, really thinking about clinical, HTA, and novel clinical designs around new and technologies and tumor agnostics. Before I start, I would like to let you know that I'm talking to you from Toronto, Ontario, Canada, which is on the traditional territory of many nations, including the Mississaugas of the New Credit, the Anishinaabe, the Chippewa, the Haudenosaunee, and the Wendat peoples, and now home to many diverse First Nations, Inuit, and Métis peoples. I want to thank all the generations of people who have taken care of this land for thousands of years. And I encourage everyone listening today to think about where they are and give thanks to the people who have been stewards of our land over the many years. So with that, let's start our workshop. And I wanted to introduce you. So for folks who may not be familiar with CADTH, CADTH is an independent, not-for-profit organization and that's responsible for providing Canada's healthcare decision makers with evidence and optimal use of drugs and medical devices. And there are a number of things that we do. CADTH delivers advice, recommendations, analysis, and evidence across a number of different health technologies, including drugs, devices, and other technologies as well. So this, and just as a disclosure, CADTH is funded by contributions from the Canadian federal, provincial, and territorial ministries of health, with the exception of Quebec. And we also receive application fees from industry, from the pharmaceutical industry, for pharmaceutical reviews and scientific advice. But this workshop is done in collaboration and planned in collaboration with the Canadian Cancer Trials Group. And the Canadian Cancer Trials Group is a national academic research group that conducts clinical trials. And there are a number of clinical trials across many different disease sites and different kind of therapies and technologies, including radiation therapy, pharmaceutical therapy, and then also many awards that folks have also, and you'll see some of the presenters today who are award winners as well for the research and the great work that they do. So before we really dig into the workshop itself, I wanted to quickly have an announcement. So for those of you who are in Ontario, Canada, we have today a special test alert, an emergency alerting system that we have here in Ontario that will decide to have this test at 12:55 Eastern Standard Time. So our workshop is scheduled to run from 11 to 1 p.m. Eastern Standard Time. So we may want to end 5 minutes early. And I also just wanted people to be aware and not be scared when this alert comes through, should we go a little bit over our time. So this is what we're talking about, platforms, baskets, and umbrellas. And why do we want to talk about this particular topic? I mean, certainly there have been advancements in genomics, and they've changed the way clinical trials are being conducted and trial designs in oncology specifically. There are a number of different kinds of master protocols where individuals are testing or groups are testing not only technologies, multiple technologies at a time, over multiple diseases at a time. And a review that was done in 2019 identified that there were 83 sort of master protocols, most of them in the oncology field, trying to understand new technologies with this new treatment paradigm or new methodological paradigm. So we're talking a little bit about basket trials, umbrella trials, and platform trials. You'll hear over this next 3 workshop sessions a number of definitions and more detail about each of them. But in short, umbrella trials test multiple targeted therapy for a single tumor type, which is stratified by a molecular alteration or genetic alteration. A basket trial tests genetic marker regardless of the tumor type. So it could be lung cancer, it could be a breast cancer, but they all have a similar kind of genetic marker. And platform trials are actually test several interventions against a common control group, which allow these interventions to enter and exit in the trial based on outcomes and some decision rules around that. So they all have their own advantages and disadvantages, and we'll get into some of that today. And they are certainly trying to understand the methods and how, from a health technology assessment point of view, from a stakeholder point of view, how can we interpret this data, and how can we think about the efficacy and the safety, accessibility and affordability. And over the next 3 sessions, we'll discuss each of those areas. So perhaps I can, we have 4 co-chairs, and since this is a partnership between CCTG, we have a number of members of the Canadian Cancer Trials Group. So Dr. Kelvin Chan, Dr. Matt Chung, myself, and Dr. Jean-Eric Tarid, who's from McMaster University and sits on one of our expert panels here at CADTH. So here's our workshop agenda and our plan moving forward. Our first session is actually discussing the past, present, and future of novel clinical trials. Our next 2, which will be coming up on subsequent days in December, we'll discuss some of the methodological issues around health technology assessment with respect to these new and novel study designs. And the last session we'll talk about talk with stakeholders about their input from an industry perspective, from a public and private payer perspective, and from a patient perspective. So now I'll turn it over to Dr. Matt Chung to talk to us about session 1.
Thanks, Nicole, and good morning, everyone. So as Nicole highlighted, the impetus for us at CCTG and specifically the Committee on Economic Analysis within CCTG was in collaboration with CADTH to really try to understand the best methods and analytic techniques that we have at this point in time at hand, with the goal of informing, obviously, future decision making. And we started off thinking about this in a framework of the past, present, and future with respect to, in the past, where we are and our experiences to date in completing economic evaluations, predominantly within clinical trials, where we are at present with regards to our current methodologies and where we're going in the future. So it was really a past, present, and future look at our group, our Committee of Economic Analysis, and our endeavors in economic evaluation in the era of precision oncology. And with that view of past, present, future, we structured today's first hour to highlight, first off, who we are as a Committee of Economic Analysis within CCTG. So we'll ask Kelvin Chan to introduce our work, and in particular with Winston Chung and Alex Louie, they'll highlight some of our early experiences with biomarker-driven economic evaluations. Then we'd like to try and understand a little bit about what's happening now with respect to state-of-the-art methodology in economic evaluations in precision oncology and some of the gaps that might still persist. And that will be framed by Dean Rege's tremendous work in this area. And finally, we'd like to look at where we're heading in the future. And we've asked Annette Hay to give us a sense of that in terms of CCTG's vision for future trial design and its prioritization of precision oncology. So just one more click, Nicole. We will be starting off with Kelvin Chen and head to the future with Annette Hay, though, just to keep in mind that today is actually Annette Hay's birthday, so we can't jump too far ahead into the future. We'll leave the second hour at the bottom there for a panel discussion, and this is really an opportunity for us all to ask questions, have a conversation, and really hear your thoughts on the challenges in this area of methodology in precision oncology. And this may not be the form in which we identify all of the solutions moving forward in precision oncology, but if we can at least state some of the problems and challenges that we foresee, I think that will really be a nice foundation for our groups, CADTH and CEA, to build upon. So with that, I'd like to introduce our first speaker, Dr. Kelvin Chan. Dr. Chan is a medical oncologist at Sunnybrook Odette Cancer Center and an associate professor at the University of Toronto. We're all familiar with his leadership in Canadian HTA and real-world evaluation, including his leadership positions at ARC, as well as clinical lead for PDRP at Ontario Health Cancer Care Ontario. In fact, he was just recognized for his roles by CADTH just this last week when he was awarded the Maurice McGregor Award as a rising star in HTA. It's a pleasure to welcome and introduce Dr. Kelvin Chan, our first speaker today.
Thank you, Matt. Can you see my screen?
Yes, we can. Go ahead.
Perfect. Okay. Thank you for the opportunity to allow me to introduce CCTG CEA, our Committee on Economic Analysis. And the focus is about currently what we are doing and what are the methods that we used in our economic analysis within CCTG. I don't have any specific industry-related financial conflict of interest to declare. And importantly, the views I express here do not represent the views of organizations that I'm affiliated with. So who are we as a group? As many of you know, CCTG has been around for 40 years. So this year we are celebrating our 40th anniversary. It started in 1980 as a cooperative oncology group. It designs and administers clinical trials in cancer therapy. And so far, this network has been made of more than 80 members, institutes, over 2,100 investigators who have facilitated more than 500 trials in 40 countries or so. And the central office is located at Queen's University at Kingston. And the overall mission of CCTG is to develop and conduct clinical trials aimed to improve the treatment and prevention of cancer with the ultimate goal of reducing morbidity and mortality from this disease. And within CCTG, one of the committee is our committee known as the Committee on Economic Analysis, CEA, not to be conflated with the cancer marker within colorectal cancer, CEA as well. And so who are we? So we have been established since 1996 because the leadership of CCTG recognized that economic factors were becoming increasingly important in decision-making, and the rapidly rising cost of cancer drugs and the modest benefits of many of the cancer therapies made it important to explore economic benefits of new therapy alongside with its clinical benefits. This is a picture of us a couple of years ago in one of our annual meetings. And so you'll probably see some familiar faces here. And a lot of them are also panelists on this workshop as well. So the purpose of CCTG CEA, there are a number of them. One of them is to apply criteria to decide whether a CCTG trial should have an economic analysis component, to develop and adapt standard methodologies to be used for economic analysis in CCTG trials, to review intergroup trials, to consider participation on economic analysis to be included in those trial design. Importantly, to facilitate the development of economic analysis skills and their application within the CCTG clinical trials community, and to interact with other health economists, cooperative groups, industry, regulatory agencies on issues of criteria for and methodologies of economic analysis in clinical trials. And last but not least, to disseminate and publish the results of economic analysis conducted through CCTG. And you can see that I've highlighted some of the purposes in red, and those I think relates to our strong desire and mission to try to use strong methods, economic methods to improve, to advance science and to collaborate with other health economists and other stakeholders. And so this workshop is perfect for that. In terms of our membership, we have two co-chairs, Dr. Matthew Chung and Dr. Nicole Mittman, who are the co-chairs also for this workshop as well. We have our senior investigator, Dr. Annette Hay. And again, happy birthday, Annette. We definitely would be celebrating your birthday. And also Patty, our study coordinator, along with many clinicians who are also liaisons with disease site committees. So for example, I'm the GI liaison within the committee. There are also health economists, statisticians, pharmacists, and lay representative. This is a schematic way of showing our productivity, our work of embedding economic analysis within many clinical trials of many site groups. And you can see that there are different trials at different phases of development. Some of those have been completed, some of those are currently active, and some of those are in active phase of development. And Dr. Winston Chung and Dr. Alex Louie will be sharing with us some of the completed work to showcase what we have been doing as an illustration about our economic analysis within CCTG. In terms of our general approach to embedding economic analysis within clinical trials at CCTG, generally we will focus on randomized controlled trials. And in addition to the data elements that's already being collected on the randomized controlled trial, we'll also prospectively collect resource utilization, which will then allow us to be able to ascertain cost of treatment, cost of diagnostic tests, outpatient visits, acute hospitalization, adverse events cost, concomitant medications cost, radiation cost, and transfusion cost. In addition to that, we also prospectively collect health utilities. Sometimes we use EQ5D, sometimes we use HUE3. More recently, we have been using more EQ5D. And by having those data prospectively collected alongside with the survival data from the randomized controlled trial, then that enables us to be able to do economic evaluations after the trial is completed. And once we have the data, naturally, we'll want to estimate the incremental cost, delta C, by computing the mean cost for each arm of the trial. And we'll usually apply discounting and adjust for censoring of data. And most of the time we take the payer's perspective, but we also occasionally will include indirect costs as well as lost productivity. And using the data we have, we also will compute mean life years with each arm and use health utilities to create QALYs, quality adjusted life years. And then by doing so, estimate the difference in the mean QALYs between the two arms. Again, we'll be applying standard discounting and methods and adjusting for censoring. And with the delta C and delta E, then we can compute the incremental cost effectiveness ratio, which is delta C over delta E. And for the uncertainty around ICER or the constant interval around ICER, usually we'll use bootstrap method. If we were to be interested in a subgroup, for example, in precision medicine, if we have a biomarker-driven subgroup, then we'll restrict our analysis based on that subgroup and then perform the analysis again and then compute ICER and bootstrapping to ascertain the uncertainty around the ICER. So that's the general approach to a lot of our analysis. Sometimes we use regression-related methods, sometimes known as net benefit regression. And some of you on the workshop may know about this already. So net monetary benefit, NMB, is defined through this formula. Essentially, we're using lambda, which is the willingness to pay threshold, almost like an exchange factor to convert clinical benefit into monetary terms, and then subtract the cost from it to create individual NMB. And then we can then compare the difference of NMB between the two arms, essentially doing a regression, looking at the incremental NMB. And the advantage of doing that is that we can then use the regression framework. And if there's any potential imbalance between the two arms, we can even adjust for potential imbalance by just adding additional regression variables. And through this paradigm, then it makes it easier to think about subgroup analysis, just like any other subgroup analysis we think about in a survival analysis paradigm by creating subgroup analysis, including interaction term between the subgroup and the main treatment effect to ascertain the subgroup effect. So that's the beauty of doing it in regression. And so Winston will be showing us some examples of that as well. Now, one problem that we have previously observed is that even for trials with pretty big sample size, here you can see that these sample sizes are not small, more than 500 patients on the trial. We can see that already the constant interval is pretty wide for the ICER. And so even with relatively large sample size, we already have a problem with precision. So moving forward into the era of precision medicine, the natural question we have is that will we be having a bigger problem of lack of precision when estimating our ICER without economic evaluation in the era of precision medicine, which it sounds slightly ironic because we are in the era of precision medicine and yet we have a problem of lack of precision when estimating the ICER. And some of the common themes and challenges that comes along with it is that the cohort sample size itself sometimes can be small, especially for economic evaluation. Sometimes the desire is to look at the specific cohort of patients with a specific biomarker and in fact in a specific cancer type treated with the intended target treatment. And keeping in mind that while in the clinical space, sometimes we do single arm analysis to try to estimate anti-tumor activity, for cost effectiveness analysis, there's no such thing as single arm cost effectiveness analysis. We always need to have a comparator, ideally a Canadian relevant comparator. So that speaks to the control group issues. Ideally, it would be nice to have a control group who are receiving treatments or standard of care within the Canadian context, but also with the same specific biomarker of the same cancer type. Ideally coming from a randomized controlled trial, but even if that were to be the case, given the potential small sample size, we may still have problems with balancing of unmeasured confounders just because of by chance with a small sample size. Alternatively, we may have to look for the control from a different source. So we may be looking at quasi-experimental design. Then we have the additional challenge of adjusting for both the measured and also the unmeasured confounder. Again, with a small sample size, the parameter we need to adjust for may be bigger than the sample size we have. And then maybe if we want to expand our source of controls, then maybe we can think about using controls from the same cancer type, but with unknown biomarker status. In that case, it would be nice to have some justification studies to show that the biomarker status is not a prognostic factor in the untreated patients with the same cancer type. And to make it a little bit more complicated, we do need to have enough information on the case as well. Sometimes we can increase power by increasing the control group sample size, but up to a certain limit. Generally, when we look at the ratio of case to control more than 1 to 4, we don't get a lot more power by increasing the control size alone without increasing the cases. So in terms of other additional challenges, sometimes we see surrogate endpoints instead of overall survival or quality of life endpoints. And it would be nice to see response rates being correlated with overall survival or quality of life, because if we are not sure, it poses additional challenges of modeling, because ultimately for economic analysis, we need qualities. Therefore, we need overall survival and quality of life to some degree. And time horizon is another challenge, that some of the trials may have shorter term time horizon to demonstrate anti-tumor effect, but with economic analysis, we need long-term horizon. So how do we manage that? What are the alternative methods to extrapolate the time horizon? So this is my last slide. So the question I'm posting here is that we probably need better methods, and we may also need more information. So when do we need better methods? When do we need more information? Generally, given the data that we have, naturally, we want the best method possible to get the best precision, the most efficient estimator for ICER or NMB. But given the data, there's a lower bound, there's a limit as to how much precision we can gain with the best possible method when we're looking at an unbiased estimator. And for the statistician in the audience, you may be familiar with the Kramer-Rao inequality and the lower bound, which is defined by the information, the Fisher information matrix. Unless we are willing to accept bias estimator, sometimes will allow us to have even more precise measure. But given that it's biased, sometimes knowledge users may not like bias estimator. That's another different discussion for the methods people. So, so how much of lack of precision problem can be improved by better methods? So I think that may be one of the themes of workshop number 1. We want better methods. We want to remove uncertainty as much as possible given the data we have. How can we do better in the future? And also, given the potential lack of precision in some cases, how much can that still be okay? How much comfort level does the knowledge user have by using some of the information when we don't have perfect information? How much of the lack of precision could be tolerated by knowledge users, such as clinicians, patients, regulators, HTA committee members, decision makers, or payers? Or in other words, how comfortable people are to possibly use information versus delay decision making by asking for more information. What's the opportunity cost of asking for more information? What's the value of information? So some of that may or may not be addressed in workshops 2 and 3. So with that, I think this is the end of my presentation. Thank you, and I will turn it over to Matt.
Thank you. Thank you very much, Kelvin. That was a fantastic overview. And we will just continue along this realm of what experiences we've had at CCTG the Committee on Economic Analysis by just introducing both Winston Chung and Alex Louie together. And each of them will provide an example of when we first kind of dipped our toe into this pool of biomarker-driven economic evaluations. So Winston Chung is a professor at the University of Calgary, a medical oncologist at Calgary Cancer Center, and has quite a few leadership roles in this realm of, again, real-world evidence and oncology outcomes. He is a member of the CCTG Committee on Economic Analysis, where his research there includes bridging the ties between our quality of life and economic committees and understanding the role of value frameworks as it's applied to cancer clinical trials. And after him, Alex Louie will be speaking. He's a radiation oncologist and clinician scientist at Sunnybrook Health Sciences Center. He holds a master's epidemiology from Harvard and a PhD in lung stereotactic radiotherapy from the University of Netherlands. And his research focuses on using comparative effectiveness methodologies in lung, kidney, and oligometastatic cancers. So I'll start with Winston.
Great, so hopefully you can all see my slides. All right. So I was asked to briefly speak about CO17 and perhaps provide a very high-level summary of the findings from the economic perspective. So just for those of you who are not in the GI treatment space, as a quick reminder, this was a trial looking at cetuximab for the treatment of metastatic colorectal cancer in the chemorefractory setting. And in the initial publication that was published about over 10 years ago, there was an improvement in survival of about 1.5 months favoring cetuximab. In a subsequent publication in the KRAS wild type group, the survival benefit was more significant and prominent, 9.5 months favoring cetuximab over 4.8 months in the standard care arm. Following this, there was also a prospective cost effectiveness analysis of this trial led by Dr. Mittman, who is running this workshop. This was published in JNCI. And again, I was invited to speak briefly about the main findings and how a biomarker-driven analysis is often very helpful. I won't spend too much time going over many of the methodology aspects that Kelvin already nicely described. Suffice it to say that many of the methods that was mentioned by him was applied in this study. So in this context, both a cost effectiveness and a cost utility analyses were conducted. This was one of the first studies, to our knowledge, that applied a pre-planned prospective data collection of both healthcare resource use as well as health utility. So for direct medical resource use, a number of items were considered and accounted for, including the cost of the drugs, physician visits, management of side effects, blood product use, as well as visits to the emergency department and hospitalizations. Health utility using the HUI index was also implemented to look at utility. The only caveat here is that non-medical costs were excluded. So unique to this study is that this approach to the economic analysis was applied both the overall study cohort as well as the KRAS wild type group separately and presented together. So I'll only have time to go through the main findings. In the entire study population, the mean overall cost was found to be about $28,000 for the cetuximab arm versus about $4,000 for best supportive care. When we look at the ICER per life year gained as well as per QALY, it was close to $200,000 per life year gained and close to $300,000 per QALY. As I already alluded to earlier, this was then replicated in the KRAS wild type group. And as you can see here, there are some notable differences. The mean overall cost per patient was about $37,000 in the cetuximab arm versus about $4,000 in the best supportive care arm. When we look at the cost effectiveness as well as cost utility, again, noticeable changes. Going from $200,000 per life year gain to about $120,000. And likewise for cost utility, moving from about $300,000 to about a little over $180,000. So interpretation from this analysis was the following, that really a biomarker-driven application of a targeted therapy can potentially produce significant cost savings. And really this sort of as an example of the value of a biomarker-driven approach. And as I already mentioned, this reflects the first analysis from an oncology cooperative group that both collected resource use as well as utility values prospectively in a phase 3 trial setting. That was a very, very quick overview of the economic analysis of this trial. I also want to bring up a follow-up study, again, involving many of the individuals in this workshop, where we also revisited the economic analysis of several trials, including CO17, where we looked at the impact of generic pricing on the cost effectiveness of these drugs. And what was interesting here is that despite the movement from branded to generic drugs, many of the values that I showed earlier still remain not overly cost effective. So I think that was a really important finding from this follow-up study as well. So with that brief overview, I'll now hand over to Dr. Louis, who will give also another example in the lung cancer space.
Great. Thank you for the introduction, and also thank you to the organizing committee for the invitation to speak today on behalf of the Health Economics Committee at CCTG. So this is a trial in the lung cancer space, and specifically evaluating a particular medication known as erlotinib. These are my disclosures. So erlotinib is what's known as a tyrosine kinase inhibitor, and so that is an oral pill that's taken and active in lung cancer. And as we now know nowadays, there's a particular biomarker that has been identified where it's more active, and that's the EGFR biomarker. And so back when this particular study was run, it was not as clear in terms of potential efficacy of this, and it was used in the second line space, whereas currently there certainly has been an explosion of developments within lung cancer from the systemic therapy point of view. So this particular trial, BR21, was a randomized study comparing erlotinib to placebo in the second line chemotherapy space. And what was found is that the median survival benefit was 2 months, comparison of 6.7 to 4.7. Going back to what Dr. Kelvin Chen had highlighted, determining when a CEA may be appropriate in the context of a clinical trial for economic evaluation. Number one, the therapy is potentially costly, which can be the case for some of these medications. And it is anticipated that new intervention may have a modest therapeutic effect to a relatively large population. And lung cancer certainly has had historically a very dismal prognosis. In addition to that, it's felt that the economic evaluation may yield important information in determining routine practice. And when we're doing such trials in the context of a cooperative group trial, it's important to look in the Canadian landscape of whether or not such findings would be generalizable, and so potential suitable number of Canadian patients. And so much like the trial that Dr. Chung just presented, this particular study also had the foresight of collecting healthcare utilization upfront prospectively. And as we can see here, the main driver of cost was actually the medication itself. And we're looking at the other interventions from a supportive care point of view, hospitalization, as well as clinic visits. It was actually quite similar between the two arms. And so very briefly looking at the main focus of the results, the delta C, or the difference in costs, was about 12,000 Canadian using $2,007. And the delta in the survival was 0.11. And so when we're looking at an ICER per year, and so this is a cost effectiveness rather than a cost utility analysis, is about 112,000. Looking at things from a cost acceptability curve point of view, i.e. the probability that the intervention is cost effective when considering different thresholds of how much society would be willing to pay per life year gained. It was considered that this was potentially a cost effective intervention. And again, going back to the central tenet of this particular symposium, that the intersection of can we select out where things may be potentially most cost effective. We know now that these particular medications are most effective in the situation where the biomarker known as EGFR is present and mutated. And this is highly collinear in individuals who are also never smokers, typically individuals who are younger, female, and Asian. And certainly these results here dovetail with that particular finding. So I'd like to thank you for your attention and hand it back to the organizing committee.
Thank you very much, Kelvin, Winston, and Alex, for providing us with a past perspective of clinical trials that look at genetic markers. I'd like to introduce our next speaker. So our next speaker is Dr. Dean Regier. He was a scientist with cancer control research at the BC Cancer and at the Canadian Centre for Applied Research in Cancer Control, or ARC. He is an assistant professor at the School of Population and Public Health at the University of British Columbia. He's also the director of the Health Economics Support and Research Unit at BC Cancer. Dr. Regier's lab, PACER, Patient-Centered Accessible and Efficient Applications of Precision Medicine, focuses on understanding access to healthcare and improving methods to estimate the benefit to our healthcare with applications to genetic and genomic technologies. So over to you, Dr. Regier.
Oh, Dean, I think you might be on mute still.
Perfect. You can hear me now, yeah?
Perfect.
Thank you.
So thank you, Nicole, and to the organizers for inviting me to talk. I actually have been promoted to associate professor and senior scientist at BC Cancer. And the idea is my presentation is going to really try to talk through clinical trial study design, data collection, and analysis that is concatenated with real-world evidence, of course, in the context of precision oncology. To do this, I will be using my experience, my lab's experience, working with clinical trials like the Personalized Oncogenomics Program in British Columbia, and also recent experiences thinking through learning healthcare systems with a view to support sustainable omics innovation. Like Nicole, before I start, I would like to acknowledge that I'm actually in Coquitlam, Canada, which is just outside of Vancouver, which is in the unceded Coast Salish territory, the traditional territories of the Squamish, Tsleil-Waututh, and Musqueam and Coquitlam First Nations. So my disclosures: in the past year, I've received honoraria from Roche. My lab is funded by Genome Canada, Genome BC, CIHR, and hopefully soon to be announced through a CFIN Kickstart program. In July this year, I started something called the Canadian Network for Learning Healthcare Systems and Cost-Effective Omics Innovation. So that is what we hope to build out, a large network focusing on the application of genomics, and that is funded through a Genome Canada GCERT grant. And then I'm also actively involved in the Canadian Centre for Applied Research in Cancer Control, which is funded by the Canadian Cancer Society. And so in terms of conceptually mapping out this presentation, the idea is I want to touch on challenges that precision oncology is facing, has faced with respect to implementation and reimbursement, and then start to do some thinking around how real world evidence or RWE methods can start to help solve those challenges. And as Calvin rightly pointed out and articulately pointed out, when we think about these high-level challenges, a lot of it is focused with respect to study design and data generation that does not meet the evidentiary hierarchy. And so as a lot of us are aware that in the precision oncology space, there is a paucity of randomized controlled trials, which of course are the gold standard, some would say, for comparative analysis. That when we talk about comparative analysis, we're talking about intervention versus standard of care. Instead, what we're seeing in the precision oncology space, and I'll give some examples of very soon, is that we see a lot of phase 2 studies being applied and used with the hope of getting not only NOCCs, Notice of Conditional Compliance, but also being submitted to CADTH BCODA, the Pan-Canadian Oncology Drug Review. And of course, you know, these are great. Phase 2, they evaluate safety, efficacy. They're typically over a short-term time horizon. They are non-randomized, and they often suffer from a lack of patient numbers. And so when we think about that in light of RCTs, the question then becomes, well, how do we think about trial design alongside the use of real-world data for real-world evidence? And when we talk about real-world evidence, the idea is that we are using real-life data under real conditions, which allows us for the potential of having greater external validity. So RCTs have great internal validity because they're experimentally designed. But the question becomes, you know, to what degree do we observe the outcomes in real life? So RWE allows us that external validity in comparative analysis. But when we talk about comparative analysis, RWE doesn't have that counterfactual that Calvin talked about. And so the question is, how do we generate that counterfactual? Even in the context of phase 2 trials, can we generate counterfactuals to observe the incremental effectiveness of a specific intervention? And so when we weave this idea of phase 2 clinical trials with real world evidence in the context of precision oncology, that's what we're calling, or at least I'm calling it, life cycle evaluation, sometimes life cycle health technology assessment, which is really trying to tease out the methods and the ideas with respect to trying to standardize real world evidence and trial data, study design, data collection analyses with a view to guide the appraisal, reappraisal, and de-adoption. So in that way, you have this not just on-off world where we say yes or no, it's a continuous evaluation when we try to think through questions around effectiveness and cost effectiveness. Now, as Nicole brilliantly put out, overviewed, and I don't need to go too much into this, there's this idea of master protocol frameworks that have emerged over the last, I guess, 2 decades now, where especially in precision oncology, where these ideas of multiple hypothesis testing in the context of the tumor agnostic conditions or tumor agnostic applications. That is, that's a basket trial where you have targeted therapies evaluated in multiple diseases with this common molecular alteration. Umbrella trials are multiple targeted therapies for one disease that's stratified into subgroups. So something that comes to mind right now is diffuse large B-cell lymphoma, and using expression to look at cell of origin and the application of BTK inhibitors. And then there's the increasingly used platform trial, which, as Nicole said, is the idea that we can drop intervention arms or introduce intervention arms during trial with that stopping or starting criterion using some sort of Bayesian framework. And here's the evolution of these clinical trial designs. So a lot of them, just as a highlight, do not, especially when we talk about basket trials or umbrella trials, do not employ randomized controlled trial or experimentally designed studies. And so we see quite a few in oncology, and we see that they are increasingly applied to look at things like clinical effectiveness. And here's some examples in the precision oncology space. We've heard some examples before, and here's some new ones. So I'm not involved in CAPTURE, but CAPTURE is a CCTG clinical trial. And this slide was given to me by Lillian and Dan. So thank you, Dr. Sue and Renouf. And the idea of CAPTURE, of course, is a basket trial where it's tumor agnostic, and the molecular profiling comes from POG, Personalized Oncogenomics Program, and I believe from Octane in Ontario. And the idea is that patients are matched by the biomarkers, by their variant, by their alterations, to a marketed but out of indication drugs, which I call off-label application. My colleagues at BC Cancer like to call it repurposing drugs. But the general idea is that you get this basket and you look at you look at outcomes like progression-free survival, et cetera. A more controversial application of a basket study that was actually put forward to CADF PCODER, I guess about a year ago, the recommendation was made, is the context of NTRK fusion, so NTRK 1 to 3, and the use of molecules like larotrectinib and entrectinib to treat these patients. And so here's the Star Trek 2 trial, which is the Roche trial. The other trial was Bayer, I believe. And the idea was you have an efficacy population, in this case of 54 adult patients with these NTRK 1 to 3 positive, fusion positive cancers, and they haven't previously received an NTRK inhibitor. And what this trial did is they had a primary endpoint of overall response rate, which was measured by RECIST, and duration of response rates. And then they had secondary endpoints of progression-free survival and overall survival. Of course, that's problematic, as Calvin said, because you have these intermediate and primary endpoints, which are relevant to the decision-making conversation, but are not as strong as something like overall survival of quality adjusted life years, which of course, when you have small sample sizes, when you have the context of a basket trial and then these primary endpoints, it makes it very difficult to deliberate on the evidence because the evidence has a lot of uncertainty around it. And indeed, when we think not just in Canada, but globally about that conversation with respect to the NTRK inhibitors, is that it's been a very difficult go for priority setting committees to really think through and balance the clinical effectiveness and think through the cost effectiveness. So on the left of this screen is the PCODER PERC recommendation, which is a full do not reimburse. And this is for larotrectinib. Entrectinib was actually pulled from the review process prior to the release of the final recommendation by PERC. In NICE in the UK, what we have is a recommendation that says, let's put this through a managed access program. So that's access with evidence development. Because we don't think the evidence base is there. We can't really make a decision, but we think that there is promise. And so what they did through the National Institute of Clinical Excellence, NICE, is they put it through the Cancer Drugs Fund to continue to gather evidence, real world evidence to look at issues around effectiveness and cost effectiveness. But I think, this case study is very instructive and a very good a way of trying to understand the shortcomings of precision oncology trials globally in the context of non-randomized controlled trial data evidence being generated. And so this is a highlight, and this is not just PCODER PERC, this is what NICE, the NICE Deliberative Committee also suggested, and these are kind of common themes, one of many common themes between the two recommendations. Being that the background frequency of NTRK is unknown, which is important for not only for budget impact, but other aspects of cost effectiveness analysis and clinical benefit. And of course, we don't know the background frequency of NTRK fusions because we don't do gene expression profile in all cancer patients or all metastatic cancer patients going through our system, mostly because it's very costly, mostly because our systems aren't set up for that. They also suggested and found quite rightly, as Calvin alluded to in his presentation, is that the prognostic trajectory of these patients are unknown, which makes it very difficult to tease out the degree to which NTRK is prognostic or the degree to which the molecule, the treatment, is actually working for patients. And we've talked about the fact that phase 2 trial was used and no RCT evidence is available, which the committee said, hey, because of this uncertainty and this lack of RCT data, the outcomes of things like overall survival and progression-free survival become very much uninterpretable, given the underlying heterogeneity, and I suggest it's biological heterogeneity in addition to the heterogeneity within each tumor subgroup. And so you have these questions, and I feel like the PCODER report wrestled with this idea quite a bit in what they put forward. Is that do we go through a tumor agnostic approach, looking at all cancers and pooling that? Or do we do tumor specific through subgroups? And what are the balances and the trade-offs of looking at everybody all together versus looking at tumor subgroups? And so with that in mind, there has been increasing calls, as we know, for the application or use of real-world data for real-world evidence. And so as in the NICE context for entrectinib and larotrectinib, there's questions become, how do we enact access with evidence development frameworks, that is collecting real-world data to inform decisions once these phase 2 trials have come to bear and we're very uncertain around outcomes. But of course, as a lot of us know, and I don't think this is a surprise, when we talk about real-world data and generating it for evidence, there's really a lack of decision-grade data and platforms. And by decision-grade data, I mean those data, how deep are those data in terms of being able to afford us the right types of information to reliably produce it as evidence to decision makers. And this really speaks to data curation and architecture. When we talk about architecture, is it set up in a way that we can concatenate all the data together? This speaks to the fact that the minimum data sets are a bit vague for precision oncology, and can revalue. Of course, Calvin's initiative is doing a great job with minimum data sets, but is there a specific minimum data set that we need to think through with respect to precision oncology, and what does that look like? And so, when we think about all these together and all the challenges that precision oncology is facing, I go from the perspective of that systems and frameworks are faced with a pretty big challenge to adapt to the precision oncology space in order to manage demand. And so we don't need to adapt because there's this idea of precision oncology generally, and we think that we like it, and it's a pretty an up and coming and force in the oncology space. So I'm not saying that we need to adapt because of that, but I think we need to adapt because we need to manage demand. And what I mean by that is not just patient demand for novel therapies, which of course is there, it's also clinician demand for novel therapies with respect to precision oncology. A lot of the times from my experience at BC Cancer is clinicians are enthusiastic about using precision oncologies in a lot of contexts because they're very concerned about their patients, and they want to offer the patients what they think is the best care, even in light of that of that certain evidence base surrounding effectiveness and cost effectiveness of precision oncology. And so in order to do this, I have a few initiatives. So that the network, as I mentioned, is called CLIO, and I'm the PI together with co-PIs Tim Hanna in Kingston and Tanya Bubela at Simon Fraser University. And what we're trying to do is we're trying to generate what I call an methodological engine room to allow for this intertwining of real-world evidence with clinical trial data. But of course, what CLIO is not doing is we're not generating frameworks. And so part of hopefully soon to be announced CFIN Kickstart program is to think through frameworks and the methods needed to support those frameworks in the context of precision oncology. And so when we talk about frameworks, and why are you talking about frameworks in terms of clinical trials and cost effectiveness analysis? Well, why I want to talk about a framework, which we'll call lifecycle health technology assessment right now, is that we have to understand the frameworks of decision-making in order to understand what type of methods and what type of clinical trial designs can support that decision-making process to produce evidence that decision makers can deliberate upon. And so this is one iteration, idea, approach to a managed access or access with evidence development framework. And the idea is to start to think through, okay, can we allow managed access or maybe conditional reimbursement for promising molecules in the absence of randomized control trials, essentially, where something like an NOCC has been identified. And then at the beginning of this framework, we can say, okay, here are the outcome requirements. Here's when we're going to invest or disinvest, or say we have enough data to make a decision. But in order to do that, we need to collect that real world evidence. We need to define which data elements we're going to collect, of course, prospectively. And then we need to concatenate that with and leverage against external data. Now, as we know in a lot of contexts, and we have these subgroups and sometimes even subgroups of patients who have, for example, an NTRK fusion or who have a specific molecular alteration. And you get these very small sample sizes. And Calvin alluded to this. What happens in the case where you have fewer patients than you have parameters to be estimated? And the question is, well, how do we address the siloing and actually incorporate this external data into our analyses? And so let's assume that we can do that, which is a tall order, the siloed data aspect. Then we need to analyze it, and not just, of course, cost effectiveness, but effectiveness and net monetary benefit, as Calvin mentioned, but also value of information. And by that, an economist means what additional value is there to collecting more evidence on the research question being investigated? And as it turns out, we can tease out the value of collecting additional data with respect to saying, hey, we can stop collecting additional data, we can keep on doing it, or we think we have enough data to make a relatively informed decision. And when we talk about should we collect additional evidence, I don't need to talk too much about this group too, but it's all about starting to address uncertainty or imprecision around our parameter estimates. And when we think about cost-effective analysis, that imprecision is not just with costs, not just with effectiveness, but that joint uncertainty between cost and effectiveness, which indeed is a tall order. So with this in mind, I'm going to talk a little bit about methods, and we're going to spend some, maybe 12 minutes talking through methods that can support some of this intertwining between real-world evidence and clinical trials. So first talk about minimum data set, I'll talk about study design, and then we'll go into a little bit of linkage analysis. So I don't want to talk too much about the exact data elements that we think and we have found that are needed to go into a precision oncology evaluation of effectiveness and cost effectiveness, just to say that we are actively thinking about it, and we are actively thinking about it in light of the fact that a lot of these analyses don't have randomized controlled trial data, so there's no counterfactuals. So we'll need at baseline and indeed before baseline, we need data on patients to reliably generate that counterfactual, which I'll talk about in a moment. We'll need to collect these data, and here's are the categories of data on the right here, at specific time points, realizing that, so something like quality of life data, of course, needs to be specific time points, where we have also the clinical data, the omic data, the utilization data happening at various time points. But we need to forget about the siloed nature of, often in my experience, the omic data is not married with the clinical data. We need to start to address that and address the siloing of data. And so what we've done is we have recruited, and we've completed, I think, 2 or 3 workshops now, and then recruited in the last month and a half in an international group of experts using snowball sampling. And what we've done is we've consented to 62, and we have 46 completed surveys. And then we're going to use a Delphi process to prioritize the data elements that we think should be collected or should be considered when we talk about clinical trials and real-world evidence. So if we assume that the right types of data are in place, how can we use that data to improve our study designs, or think through these study designs rather, that allow us to generate counterfactuals, that is, mimic in a quasi-experimental way standard of care when that hasn't been generated through a randomized controlled trial. And so here's a conceptual study design that we're applying in PREDICT, and that we hope and that we will be applying in future states through initiatives like the Marathon of Hope Cancer Centers Network. And so the idea is that, and this is the top half here, is that we have over time the ability through, for example, a clinical trial and part of PREDICT is a clinical trial that looks at biomarker positive patients and biomarker usual patients that have gone through treatment and usual care. But of course, they're in the intervention arm because part of the intervention is not only the molecule or the treatment itself, it's the intervention of the comprehensive genomic profile, the next generation sequencing. So we have to think, when does the intervention start? We also have the idea of a matched cohort where biomarker status is unknown and these patients have received usual care. And that matched cohort is going to be experimentally designed. It's going to be prospectively collected. Also, we're going to draw on our existing databases. But of course, it's difficult because we don't know the biomarker status. And the question becomes, as Kelvin pointed out, are we generating quasi-experimentally designed counterfactual, which can be useful in understanding what would happen in absence of next generation sequencing. And then, as I said, leveraging this external data is very important. Leveraging external data is very important, especially in the context of clinical trials or external data sets that have the information or the data of biomarker positive usual care and biomarker negative usual care, which will allow us to get a bit more depth, not only increase our sample size, hopefully, but get a little bit more depth with respect to answering that question of that, can we generate a matched cohort where biomarker status is known through these quasi-experimental analyses. Of course, the big question is, what type of analyses can support this type of evaluation? Is it acceptable to decision makers, and what kind of evidence do we need to generate in order for it to be acceptable to stakeholders? And so I'm going to draw a little bit about on my experience with POG, the Personalized Oncogenomics Program, and really start to highlight some of those data sources and linkage and analyses that are needed to support these kind of study designs and thinkings around using real-world evidence and clinical trial data. And so in terms of data sources and linkage, this is one iteration of this. So this is POG. And what we were able to do is we were able to link BC Cancer Registry to external data holdings through Pop Data BC to get that full measure of health resource utilization, together with things like overall survival, pharmacy database, pharmacy use, etc. This is data sources and linkage in PREDICT, which is a bit more difficult because what we're doing in PREDICT is we're trying to generate a system that is ready for generating real-world data for real-world evidence. And so we need to, on the left, we have PREDICT components. So we're implementing focus panel testing. We're implementing data capture, prospective data capture, both on the clinical trial side and across BC Cancer through something called EPIC. And then we're trying to use external data holdings from Flatiron Health and the Rosley-Czech clinical trial data and then put that all together with a view to answer questions around cost effectiveness and effectiveness, but of course understand that there will be missing data, and how do we address that missing data through things like data science. In my last few minutes, I really just want to touch on study design and the analysis to support that study design where counterfactual doesn't exist. And so this is something that we've just published on using machine learning, what amounts to supervised learning, which is an algorithm matched based analysis. And so in general, I'm sure like a lot of folks in this room, this virtual room know that, we have this idea of matching methods, which enable the identification of a counterfactual where none exists. Challengingly, though, the validity of these approaches really rely on the underlying assumption that after matching, there are no unobserved differences remaining across treatment groups, which we term ignorability. But we have to assess ignorability, and we do this through enumerating the balance of the entire covariate distribution. So not just the means, but also the underlying distributions or background distributions that these means follow. And any residual balance, of course, means that we are not doing a very good job in our propensity score matching. So we have this idea of propensity score matching, which is being used a lot and is a great method. And the idea of a propensity score is that you balance the score, and after matching on the true score, this idea of ignorability will be met. In reality, however, it's impossible really to know if a propensity score has been specified. And what we do as researchers through propensity scores, we apply an iterative process of manually modifying the background model specifications in order to support the idea that we say, okay, we have just ignorability and we have this valid inference world. The problem is that we are not very good at doing this on a manual basis. And even after like hours and months, we still might not get a good counterfactual. The idea of machine learning or supervised learning is really that it automates this through an algorithm-based approach to really converge on the best or what's optimal really matched cohort, thus producing these least biased and hopefully more statistically efficient estimates. And so I have 2 slides left. And so here's what we did. So we were really interested in this machine learning based approach. And so this is POG patients, and this is the first 230 POG patients. And what we did is we looked at our unmatched cohort, a propensity score matched cohort, and this genetic matched cohort, which is what the machine learning approach is called. And on the left, we have all subtypes, the tumor agnostic context, where we can see there's no difference, and this is Kaplan-Meier survival curves. There's no difference in intervention versus standard of care. So that's POG patients versus POG naive patients. And those POG patients, of course, include those who did have treatment guided by whole genome transcriptome analysis versus those who did not get treatment based on their molecular profile. In the middle, we subdivided into those who did get treatment based on the WGTA, whole genome transcriptome analysis information, those POG patients who actually weren't able to get drugs, and then our standard of care cohort, which we called POG naive. And what we found is that in the unmatched cohort, there was quite, and actually indeed in the propensities and genetic match cohorts, there was signal there in terms of benefit for those who received POG guided care through whole genome transcriptome analysis, those people who were actually able to get drug. And then we also looked at the tumor specific. So we looked at breast cancer patients specifically. In the unmatched cohort, the POG naive, those non-POG patients were doing quite a bit better than POG patients, which was interesting. Once we started applying the propensity score match cohort, those lines converged a little bit more, but there still seemed to be some benefit. And interestingly, in the genetic match cohort, that all that benefit between intervention and standard of care went away. And I highlight this because it's important and it's instructive to say that our approach to matching, to generating those counterfactuals will influence our parameter estimates and could adversely influence our conclusions. And so here's my last slide. So in summary, I know that was a lot of information. In summary, the highlights, if I could review, is that we need to start to thinking through this tumor agnostic versus tumor specific world. We need to be specific about the intervention. We need to be specific that it's just not, in the case of precision oncology, intervention is just not drug. It's also the actually technology being used to identify the biomarkers. We need to really think through our counterfactuals and the methods to support the counterfactuals and how RW can inform that. And then I'd also like to round off by saying our priority settings frameworks, I think, will evolve in the face of precision oncology. The question is, are we ready with the right types of methods and the right types of supporting data in order to support high quality evidence? And then finally, I wrote this just last minute as Kelvin was talking. I think we need to embrace uncertainty and then think about, if we don't embrace it, we need to start to resolve it through things like valid information and additional evidence generation. So thank you.
Thank you so much. That was, I would say, literally mind-blowing. I'm feeling lightheaded and in a good way. I'm sure many of you have questions. I think I have 100. But we have purposely saved the question period and discussion period for the end of the talks. And we do still have quite a lot of time to be able to kind of explore. So, so please do save your questions, or if you like, you can also type them into the Q&A box and we will certainly get to them. But first, we would like to introduce our last speaker, Annette Hay. Annette is an associate professor at Queen's and a senior investigator at the Canadian Cancer Trials Group. Her research focus is clinical trial design, methodology, including health economic analysis and data linkages. And she is a malignant hematologist at Kingston Health Sciences Center. She is perhaps single-handedly advancing both the science and actual manufacturing of cell therapies in Canada, including CAR T type therapies. She's a mentor and a great friend of mine. Welcome, Annette.
Thank you very much, Matt, and everyone. Can't think of better people to spend my birthday with. And there is nothing I am doing single-handedly. It's with a great team that I've no doubt will continue to expand. Disclosures. In my role as a senior investigator at the Canadian Cancer Trials Group, I have received research support from various pharmaceutical companies. And this was actually quite a tricky talk to prepare for. I thought it might be helpful to go right back to the basics just to define exactly what we're talking about. Precision medicine is using information about a person's own genes or proteins to prevent, diagnose, or treat disease. And in cancer, that usually refers to specific information about a person's tumor. An example would be trastuzumab for HER2-positive breast cancer. Tumor-agnostic therapy is taking this a step further, using drugs or other substances to treat cancer based on the cancer's genetic and molecular features with no regard to the cancer type or where the cancer started in the body. So it's using the same drug to treat all cancer types that have the genetic mutation or biomarker that's targeted by the drug. And an example would be pembrolizumab, the PD-1 inhibitor, approved by the FDA for individuals with MSI high who have progressed after prior treatment, regardless of their PD-L1 expression, regardless of their cancer type, tissue type, or tumor location. So predicting the future, pretty tricky. I've decided I'm going to demonstrate the future that I wish for and really use this just to frame questions in my mind that perhaps we can address during the discussion period and in future sessions. One thing that will not change at the Canadian Cancer Trials Group over our next grant cycle, 5 to 7 years, is the mission. It remains to develop and conduct clinical trials aimed at improving the treatment and prevention of cancer with the goal of reducing morbidity and mortality. And if you're feeling snoozy and want to fall asleep, just glance through this slide and it's the next 10 minutes. Firstly, I believe that basket trials, umbrella trials, and platform trials, as have been really nicely presented by Dean, will remain. What will change is that we're going to need to think about how to approach them more cleverly. Now, as an example, in development, this is a pretty ambitious initiative being led by the National Cancer Institute in the US. It's called the MyeloMatch platform trial. There has been, over the last 2 to 3 years, extensive involvement by all of the cooperative groups across North America to build a really robust platform for all patients presenting with acute myeloid leukemia and myelodysplasia across Canada and the US. It starts off with laboratory testing, a master screening protocol whereby, for example, a patient's bone marrow can be assayed to identify specific disease factors. In addition to that, patient characteristics and clinical factors will be linked to determine which group of studies they best fit into. There are specific cohorts for older AML patients who are not fit for intensive chemotherapy or who are fit, for the young adults, and for those with myelodysplasia. And just to give you an impression of the complexity, focusing just down on the bottom right, this is what this looks like. So step 1 being the screening protocol with karyotyping and mutation analysis. From there, patients will be identified and invited to participate in one of 4 randomized clinical trials, those with high-risk tumor features, good risk features, and others. And each of these studies is being led by a different one of the cooperative groups. In Canada, we are leading the others in red. Thereafter, after induction therapy, depending on one's response, whether there is minimal residual disease positivity or negativity, patients will be run into the next trial, looking at options to improve outcomes there, and thereafter, a third maintenance trial. Now, I'm not exaggerating, this has been 2 to 3 years of work by many. Of course, there are many complexities as you think through how an individual patient will cycle through this, and to be sure each individual will get the best care possible, how we will link the data, how we will ensure that it's interpretable at the end, knowing there are multiple possible permutations, and how this will inform next steps, either changing practice or informing subsequent studies. Biomarkers are absolutely crucial in an era of precision medicine, and particularly considering tumor agnostic therapy. And this is where I am grateful for many good, clever friends, including those who are very familiar and comfortable with biomarkers in the laboratory. And one of these friends is Harriet Fallotter, who some of you may know in Kingston, a molecular geneticist. And in the same way that we take a new therapy and run it through testing from laboratory to clinical trials, phase 1, 2, and 3, a broadly similar approach needs to be taken for biomarkers through from their discovery, evaluation, to potential implementation and impact of those that add value for patients. We need to figure out how best to evaluate the clinical utility, validity of both clinically and analytically. And right now, there are limitations in our ability to do this, even in disease-specific indications. This gets even more challenging in tumor-agnostic settings. Just because a biomarker works in 3 tumors does not mean it's going to work in the 4th. So with Harriet and others, we are working together to get expertise to help inform design of our future clinical trials involving biomarkers, and also liaising with the Ministry of Health to help them identify what's important to look for when deciding which biomarkers will be appropriate to try and adopt and implement at a population level. Recognizing resources are not unlimited, and we have to be sensible both in our testing and determining priority biomarkers for implementation. An example, the LY17 clinical trial currently open at the Canadian Cancer Trials Group for people whose lymphoma has come back. This is a rolling platform trial. The control arm, RGDP chemotherapy, is open to accrual at all times. Our currently open experimental arm is RDICEP, the goal being to pick a winner in this randomized phase 2 trial to then move forward to phase 3 testing. There are extensive laboratory correlative studies built into this trial, including DNA, cell of origin, immunohistochemistry, fluorescent in situ hybridization, and others. And this laboratory biomarker work is being driven by Led Scott and colleagues out west. Now, what we have learned and what soon will be publicly available information, the manuscript is close to ready, in the ibrutinib containing experimental arm, which is now closed to accrual because it did not meet the first interim futility analysis. We now understand, having conducted all the correlative samples and link this biological information with the clinical outcome data, that actually now you can see how many of the people who enrolled on that arm, perhaps you could now predict would not respond well to that therapy. Now, at the time the trial was designed, we did not know that. We certainly were not ready to incorporate that up front, but it certainly does have rationale moving forward to consider incorporating biomarker studies earlier, recognizing that brings logistical and financial challenges. So in the same way that we develop therapies through clinical trial testing, there also needs to be an iterative, collaborative process to linking biomarker testing with clinical trials. Precision medicine brings a huge amount of big data, as Dean has very clearly alluded to. both from whole exome sequencing and other big data sets generated by laboratory studies, and also as we try and bring in other data sets to improve our efficiency in conducting clinical trials and the outcome data we collect. So we need help in people who know how to handle it, from AI to computer science to IT, and managing all of these and pulling them together efficiently and safely, with input from patients to be sure that we're addressing their potential concerns about privacy. My next 2 points are about cell therapy. June Carl predicted in 2020 the next decade was going to be the decade of cell therapy. And who am I to disagree? I wonder if cell therapy is going to be an example of where it may be time that the tide starts turning. And while we've been working towards making therapy more and more personalized, maybe the time will come that we can start to depersonalize it slightly. And also, as clinical trialists, now with these disruptive technologies emerging and new approaches, I think it falls to us to start to expand our usual networks to include those expert in cell manufacturing and incorporate alternative approaches into our clinical trial testing. Many of you are likely familiar with this, but chimeric antigen receptor CAR T cell therapy in brief, super cool. And for some patients with leukemia and lymphoma, where all other treatments have failed, bone marrow transplant, chemotherapy, radiation therapy, this appears to be leading to long-term remissions and perhaps cure. The current process is that a patient's own blood cells are collected. These are then shipped to a central manufacturing facility. If you're in Canada, that's currently in the US. There, the cells are engineered so they recognize an antigen on the individual's tumor. Right now, CD19 is the antigen most developed. Then these cells are multiplied and eventually given back to the patient. The limitations: the cost is daunting. The side effects can be very serious and in some cases fatal. And the process outlined on the left can take about a month for a patient to have to wait. And for individuals with these serious aggressive illnesses, sadly, sometimes a month, they just don't have. Now, there are alternative potential processes that could build on this exciting work. Local manufacturing using closed manufacturing systems may be twice as fast and up to 10-fold cheaper. This technology is being tested in Canada in the CLIC-1 clinical trial, which is open. Off-the-shelf allogeneic products are in development, such as using natural killer cells. And these sorts of products, which would be in a way depersonalized, you could still have a product that targeted CD19, but could come from a separate donor who could provide a batch rather than having to wait for uniquely individualized therapy, may simplify and may expedite availability of treatment. But all of these alternative processes need rigorous testing. And to do this within Canada, there is a recent developed collaboration, Accelerate Canada, that I'm pretty excited about. This brings together a fantastic network of experts from across the country and with expertise from outside of Canada, along with industry collaborations. The platform takes it right the way through from discovery of new technologies, new targets, through to those with expertise in various approaches to manufacturing of cells, through to rigorous multi-center clinical trial testing with strong laboratory correlatives, and ultimately, given that the goal is to identify and prove new effective therapies, deliberately includes policymakers, both from CADTH and Health Canada, to ensure that we can get these new products that are demonstrated to be effective to patients quickly. It's complicated, hence there are various different individuals who bring different aspects of the expertise required to do this, knowing that no one person has all the required skill sets. But through this collaboration, We will innovate manufacturing and test it rigorously, including testing of next generation cell products, and importantly, create an economic framework designed to inform health policy, bringing not just the usual health economics that we are used to doing in this group, but also expanding to things like value stream and supply chain. As I suspect all of you know, prospective, unbiased, holistic approaches to health economic analyses remains very important, as outlined in several previous talks, including Kelvin's. Now, we've been good at doing cost effectiveness analyses, cost utility analyses, and we should keep doing those. But I think we need to expand, considering, for example, biomarkers, looking at the sensitivity and specificity of different assays and tests, looking at the costs of doing them and the costs of obtaining the specimens on which to do the testing, to provide a more holistic approach that will be more informative at a policy level when considering the whole healthcare budget. And ultimately, with the intent being to improve patient outcomes through informing policy, we have to engage decision makers. And so I'm pretty excited to be part of this first workshop and see where the conversation goes as we work together to ensure that as we design clinical trials, they are designed to meet the needs of those who will be using the data generated by them. So I believe the future of clinical trials in a tumor-agnostic era of precision medicine requires joined-up thinking and broader collaborations than we have previously been most comfortable working with. Patients always have to be the center. They are the people who step up willingly to be part of research, provide specimens on which we can do biomarker testing, and must continue to be engaged as the ultimate beneficiaries. But bringing in meaningfully laboratory scientists, clinical trialists, data scientists, and policymakers. So with that, I think we're probably into question time and panel discussion, and I'm interested to see where this goes.
Thank you so much, Annette. And again, thank you to all of our speakers. That was extremely stimulating, and I think it has already generated quite a few questions within our chat box. Feel free to, again, continue to use that chat box to type in Q&A questions, or I believe, Nicole, I might be right on this, that people can actually ask questions as well to the panelists. Maybe I'll start off with a question that is within the chat box, and Dean, maybe I can start with you on this. You did talk about decision grade evidence as it relates to perhaps more clinical information, but how do researchers, I guess, try and obtain that same decision grade level of evidence for other aspects that we input into our analyses, in particular things like utilities, as well as even decision thresholds for ICERs?
Yeah, no, I think that's a really good question. It's something, actually, a question I hope would be asked. And I think what we see is that so I think about decision grade data for evidence being having the right types of data to support the types of analyses that decision makers are asking for when they deliberate on questions of efficacy, effectiveness, and cost effectiveness. And, with the idea that, do we have decision grade data now? I think the answer is, in the prospective setting, when we have a clinical trial, we can, if that minimum data set is well thought through. If we collect the health-related quality of life, the preference-based measures at the right time points, and then also concatenate that with the clinical, the genomics, and the health services resource utilization for this. I like net monetary benefit estimate of cost effectiveness. But I'm afraid for the real world evidence context, we don't. And so what we see at BC Cancer, what we see, I think, to a degree in Ontario and maybe to a lesser degree in Alberta, is that is that all the types of evidence
I mean, you spoke a lot about silos, even just, as it relates to these patient tumor types or biomarkers, but certainly, our classic world of clinical trials is somewhat siloed and distinct from, real world evidence collection. And I guess what you were seeming to propose in terms of this architecture is this kind of, I don't know if it's really almost like a seamless collection from the trial level all the way through to the real world aspect of data collection. And that makes a lot of sense. And it makes a lot of sense in terms of even efficiencies at the trial design level. And then that maybe you could speak to this as well. But, if we knew that in the real world setting, stuff will be collected, maybe in the trial setting, we don't have to collect as much, granular detail. And then similarly, at the end of the trial, you might be able to, ascertain that perfect information that you still need to obtain. But is that actually being done? Is this what you're thinking of in terms of this kind of seamless entire package of data collection?
You articulated it better than I did. I think that is like the hopeful future state of analyses alongside clinical trials with clinical trials of the incorporation of real world evidence. We talk a lot about the burden on our trial nurses, on everybody in collecting and inputting the data. And the question becomes, in a future state, can we have a healthcare system that allows us for this real-time curation of data that takes some of that burden off of those already very busy individuals who are chasing up clinical trial data? And so, I mean, that's the future state hope.
Yeah, and then could you comment on that? Like, is that something we're kind of open to, and kind of it would perhaps involve considerations of what the real world aspects of data collection will be, even at the time frame of when we're designing these earlier phases of studies?
Absolutely, that is the intent and a future vision. So it's exciting to hear the work that's going on in BC as well. And we have done several pilot projects within the Canadian Cancer Trials Group, linking with ICES, linking with Cancer Care Ontario, linking with Statistics Canada. So there's definitely opportunity to improve upon that and try and identify a more efficient method to do it rather than multiple agreements with multiple different organizations on a trial-specific basis.
That's excellent. Thank you.
Matt, Nicole here. I'm just wondering if I could just add something. When this Committee on Economic Analysis was first formed, and we used to be a working group more than a decade ago, we used to ask for everything. And we were promptly not scolded, but told that, the compliance with completion of all of these questions, it is very burdensome, not only for, as Dean mentioned, the nurse coordinators, for example, or the study coordinators, but also for the patients. And they're particularly asking questions outside of a primary outcome, right, which would be a clinical outcome, and asking things about, did you go to the eMERGE and what happened to you, for example. If there's an ability to streamline that, and we've been working, I think, over the past decade to do that, and it sort of culminated with Annette's work of linking the clinical trial data into some of the administrative databases that exist. And, would it be great that every province and territory was connected? Correct. That's the ultimate vision, that even the genetic information isn't on someone's personal laptop. It actually is in some sort of government or publicly funded, accessible endeavor. That would also be something to look forward to over the next little while. But there certainly is a movement afoot, right? We've talked about this, that, if a clinical trial runs for 60 days, at day 61, you don't know what happens. So even some of the governance structure around, and Annette has done some of this pioneering work, is asking for consent to link your data at the same time as you're asking for consent as being part of a clinical trial, asking for consent to follow up with patients. And I would say about 10 years ago, ethics committees used to say, well, the only way you could do that is if you receive personal health information. I think over the last few years, and Annette can attest to this, they've certainly opened up with being understanding about the purpose of collecting personal health information so that there could be further follow-up, whether that's in-person follow-up, whether it's phone call follow-ups, whether it's linking into administrative data. So the world has evolved, much like we've gone from very simple designs to very complex designs. Oh, you're on mute, Matt.
Thanks, Nicole, for that. I'm also just going to go back to a couple of the questions, and I might paraphrase them. This might come back to you, actually, Nicole at Cardiff, or others at Cardiff. And it's really a question around comparing the environment in Canada to the UK and other, I guess, jurisdictions that rely on health technology assessment for decision making. But they use the UK as an example around the entrectinib and larotrectinib agents because the UK specifically allows for conditional access and reimbursement or reassessment, sorry. And I guess the question is, I guess specific to Cardiff, is there, I guess, is Cardiff on board with this type of, I guess, conditional reimbursement and reassessment, that type of cycle? Is there guidance that we can get from Cardiff on this real world evidence component?
Yeah. Yeah, thank you. I mean, I can try and answer that. And I think, understandable question, and thank you for the question. I mean, certainly we do have Cardiff have a reassessment framework as well. And so we're actually working with our provinces and territories to help understand, what would that entail? So the example would be if we continue to generate evidence, who generates that evidence? Is it the provinces and the territories? And we just talked about burden. Who pays for that evidence generation? What kind of evidence are we looking for? If we think about it from a reimbursement point of view, Cardiff does already accept real world data. Anything outside of the clinical trial space, actually, we could consider broad to be real world data. So utility data often, or health preference data, can come from that source. Comparative data, utilization data also comes from real world evidence sources. The challenges around the tricky and the stickiness is around the efficacy piece, right? Safety data can come from registries. And so I think the challenge that we're trying to look at as well is, what structures do we need? What kind of criteria do we need? What methodological expertise do we need? So we're not quite there yet, but we're certainly thinking about this and working with other jurisdictions to understand how they've done it.
Great. I think, Winston, did you want to come in as well and talk a little bit about your experience in Alberta with regards to, I believe, learning health systems?
Yeah, I think there were a few comments earlier about one of the issues of RWE is the time lag. So oftentimes when an event happens or an outcome happens, in most jurisdictions, there could be a lag of a year or two, if not more. So in Alberta, there are now discussions about creating learning health systems so that we can get more real-time data. Because I think that's kind of part and parcel of the current conversation, because one of the issues why perhaps HTA bodies are a little more reluctant to adopt RWs because it's not, by the time data is generated, it could be 2 or 3 years out. So I think that's a major issue. So I think getting data and outcomes at the point of care, for example, is a topic of major interest in my center. I know there was a question about Epic and new EMRs, for example, how we can leverage that. So we're definitely doing our best to look into it to see whether we can decrease or narrow the time lag, because I think that's a major point of concern, I think.
Thanks, Winston. I'm going to just keep going through some of these questions. So there is one about going back to some of the randomized control trials that we would typically see in personalized medicine. And although maybe the perception is that there's a lot of focus or discussion right now on personalized medicine, there is actually few executed or completed randomized trials And maybe, Annette, I might ask you to speak to this a little bit, but is that your perception as well? And is there a perception that there's a lack of funding or support for these trials? Or is it coming from more the clinicians or the researchers at the CCTG level, and perhaps the perception that these types of trials are not as essential?
That's a huge question, an important one that could approach in several different ways. I think the questioner actually kind of addressed the answer. These are really hard to do. And certainly at CCTG, there are personalized trials we have tried to launch and actually not managed, partly because of the complexity of bringing in drugs from multiple different pharmaceutical companies, of ensuring you have access and funding to standardized biomarker or diagnostic testing to ensure you're putting people in the right cohorts. So we certainly do have successful examples, such as the CAPTOR PM1 study that Dean alluded to, and that's working well, which is really good to see, but even that is not randomized. So I think one of the issues is the logistics. The other is particularly when so many of the biomarkers are pretty low in incidence, and certainly there are trials running for biomarkers or predictors that may only occur in 3 to 4% of the population. That is a huge amount of screening to identify a small number of people who may be eligible to participate. So one of the other ways that I think we need to think about how to improve that is, for example, Harriet Falotter has developed for NTRK a screening test that's fairly cheap. So she can then figure out the population of people who are more likely to have the mutation, and then do the more expensive screen testing on them for that specific abnormality. So I think those are two of the issues, and I'm sure others could expand to include more. There's not a lack of willingness. It's figuring out how to do it properly.
And is your sense that it needs to be done because these are such rare, I guess, subtypes, as well as these trial designs are so complex that it potentially needs to be done collaboratively? Are we doing these typically with international cooperative groups? But then the risk of that is, do we lose kind of the ability to contextualize the information for Canadians? And again, this also maybe leads a bit towards asking Dean, like, do we also have the ability to kind of share real world evidence that other jurisdictions have already looked at, or is it less relevant to Canadians? Certainly, I appreciate we're just north of a behemoth in terms of the US, but there, I don't know if they have as much of that interest in real world evidence collection, if a lot of their decision making, essentially their FDA approvals, essentially comes with a rubber stamp of drug funding. So is there as much interest in that level of post-marketing data collection? So sorry, this was somewhat meant to ask both of you your thoughts.
Well, maybe I'll quickly chime in. So I think in the context of the United States, there are healthcare systems in the US that do an amazing job of curating real-time patient-level data and making decisions based on Intermountain Health comes to mind immediately. I think in terms of Canada, I think we have siloed data systems. Even within investigators are reticent to share their investigator-led data with those who could use it to inform effectiveness and cost effectiveness. And so I think that's one silo. I think across Canada, across jurisdictions, because there are different data governance laws and research ethics in place, without that prospective consent, as Annette talked about, it becomes very, very difficult, if not possible, to share data in the hope. But again, I'm going to appeal to a future state where I think that, through ideas about, federated data analytics, so using the data doesn't leave the jurisdiction, but is consented to have somebody with permissions to analyze that data in BC if they are in Ontario. I think that is a promising future. I think that we are seeing that now with initiatives called, like, for example, CanDig, which is Stephen Jones, and that allows for federated and demonstrates federated data analytics can happen. But that's just in the omics space, right? We don't have any of these great databases in the federated data analytics world that concatenates all that omic data, clinical data, economic data, quality of life, all of that is not in one place. And I think, one of the initiatives like the Marathon of Hope Cancer Centers Network through the Digital Health and Discovery Platform initiative could get there in the next 5 years if they indeed start to collect the right types of data at the right time points and have the correct permissions in place and realize the value of health technology assessment in addition to the omics, in addition to the clinical outcomes.
Annette, any thoughts from your end?
Ditto. Love the passion. Yeah. It's a shame we can't hear from the people on the line as well, because I think there's a shared passion and vision here. It's just figuring out how to execute it.
I'm just going to read a question from Pierre Villeneuve from Ottawa. He describes that there's a lot of heterogeneity in some of the available diagnostics. And again, Dean, I think you spoke to this in terms of considering not just the intervention, but actually the actual test system itself. So he speaks about the complexity, uncertainty in introducing just the actual diagnostic panels themselves within the HTA assessment. And I'm thinking he's asking a little bit about how we address the actual aspects of the complexities around the diagnostic tests and panels. I guess maybe issues like sensitivity, specificity, but also, thresholds of diagnostic test positivity, as well as even just availability, cost. But how do we incorporate that in terms of into our decision making, I guess?
I mean, I think that's like spot on. We need to incorporate that into decision making, and there are a number of different methods in our toolbox that we can use. We can use traditional methods would be to use decision analytic modeling to adjust the sensitivity and specificity and to build out across the entire patient journey or trajectory the implications of those different thresholds. I think we're developing methods in terms of the regression analysis to allow for different sampling procedures or oversampling or using inverse probability weighting that will allow us to do that. But I think that this, why this is especially important is because we are using different genomic approaches, uses of next generation sequencing, comprehensive genomic profiling, whole genome, whatever the technology we're using, this will have implications in terms of what we're finding in terms of the underlying molecular biology of patients. And we need to address that in our analyses and then control for it or look at sensitivity around outcomes depending on the application. Is it a focus panel? Is it the homebrew OncoPanel that we use in British Columbia? Is it whole genome transcriptome analysis? All that will bear on the intervention, which again, as I said, is both the technology itself and also the downstream treatments, the drugs that are being used.
Agree, and it's a good question, Pierre, and maybe something we could reflect on more. So for example, in the MyeloMatch master platform that I presented, there is an entire committee who have been contemplating which are the appropriate tests to do, recognizing in the trial the funding will be provided by the NIH for these studies, but we still had to think very carefully about how can we justify the best tests to get the most information out of whatever we use. But what we have not done is thought through how we could look at the sensitivity, specificity, cost of testing, and the value of information from that. Right now, I don't think any group is focused on that, and maybe it's something we could think about putting forward to build in and learn from that particular large study.
Excellent. Well, I'm just conscious of the time and that we're 5 minutes away from this Ontario alert. So what I think we might want to ask is if any of our speakers have any actual final thoughts. Obviously, this has been a very stimulating discussion, and it's actually quite exciting to understand that or to see that kind of all of us in our different realms might be able and very interested in actually working together with this common vision of really creating the best decision that will inform decision making in the future. So I don't know if, specifically Annette, Dean, unfortunately, sorry, Kelvin had to leave a little bit early. He had a conflict as well. But Annette, Dean, Alex, and Winston, any final thoughts from your end or next steps that you would actually see as being ways that we can, again, either collaborate or work together towards this common goal?
Who do you want to go first, Matthew?
Well, your mic is on right now.
Okay. I think so. The trainees in my lab always, when I talk about future states and get very excited about it, they say I kind of sound like Santa Claus in terms of like, this is so amazing, we can do all this great thing. I'm not. I guess we can't be ignorant to the politics and the challenges in our healthcare systems about real world evidence concatenated with trial evidence. And so we have to be a realist about it. On one hand, I think that there are opportunities to build out infrastructure, architecture, and curation to help promote some of the methods that we are all really passionate about. But I also think that, again, there's going to be this conversation around frameworks and which managed access frameworks will be acceptable to decision makers, whether it's at CADTH, whether it's in British Columbia, Ontario, Alberta. There needs to be that conversation between CADTH and jurisdictions about managed access with a view to generate the RWE, and what does that look like, and what is acceptable to stakeholders. And then I think that the reason why I think I'm passionate about it is because so I'm an economist. My background is just economics, and so. The reason why I'm really interested is I'm actually what's called the demand side economist. I'm really interested in that kind of clinician and patient driving for new interventions that are based on precision oncology. And I think that systems, as they always had, but specifically now in context of non-RCT evidence driven data, I think that there's going to be this increasing demand, and that demand is going to have to drive differences or new frameworks. And I think CADTH, VCODA, and that PERC recommendation for LARO did a pretty good job of like articulating that we are struggling with these challenges, and we need to start to think through the methods to support the addressing of these challenges.
Yes, very succinctly, thank you to CADTH and the organisers for putting together a really good session, a really good panel. It feels like the start of an important conversation that's going to continue.
So maybe I can just jump in and 1 minute before our emergency alert testing system in Ontario. Thank you very much to all the speakers. I think you've managed to stretch our minds and thinking about a complex issue. And the word I would walk away with is flexibility. We need flexibility around trial designs, flexibility around technologies, flexibility around methods, around data, around policies, and even around the infrastructure. And so we have a lot of work to do. So session 2 and 3 coming up will approach some of those issues. So session 2 we'll talk about methodological issues from an economic perspective, from an ethical perspective, and from a clinical perspective. And session 3 we'll talk about talk with stakeholders and their thoughts on tumor agnostics or these kinds of trial designs. Again, thinking about it from a patient perspective, from a payer perspective as well. So, and a clinician perspective. So we hope you will join us as well. And we hope that the audience has really been able to stretch their minds with respect to this work. And thank you again to our presenters. With that, we'll close the session.